So welcome to our Catch the Next Big Wave Symposium. I'm Jane Moores, Assistant Vice Chancellor for Technology Transfer at the University of California, San Diego. I'm very pleased that you could join us for this discussion of the future of biomedical research and innovation. Like the rest of San Diego, we were delighted by the news that the 2014 International Bioconference would take place here. We saw at once that this would give us an opportunity to host a lively exchange of ideas and perspective from a global view. San Diego and our UC San Diego campus are home to scientific trailblazers from around the world. So we know that biomedical innovation flourishes when we bring together the best minds from across nations and disciplines. Today, four of UC San Diego's most distinguished innovators, a stem cell researcher, a neuroscientist, a bioengineer, and an astrophysicist turned computer scientist, will look into the future of biomedical research to envision the next big wave of trends and developments. Our moderator for this discussion will be Steve Fallon, intellectual property attorney with Greer, Burns, and Crain, one of the symposium's two gold sponsors. Our other gold sponsor is Perkins Coie, and our bronze sponsors are Gavrilovich, Dodd, and Lindsay, Kilpatrick, Townsend, and Stockton, Shu Chien, and Sutherland, Aspel, and Bryan. We're very grateful to all our sponsors for their generous and enthusiastic support of today's symposium. I've also been asked by Sandra Brown, UC San Diego's Vice Chancellor for Research, to convey her best wishes and her regrets that she couldn't be here in person because she had a prior out-of-town commitment. She particularly wanted me to emphasize how pleased the UC San Diego campus leadership is by the increasing interactions between academia and industry. This event and your presence here exemplifies that very positive trend. And it's now my great pleasure to introduce our first speaker, who has been a champion and a catalyst for technological innovation for nearly half a century. Bob Dynes is a physicist and leading authority on superconductivity, who has served as both Chancellor of the UC San Diego campus and President of the University of California System. Bob's numerous scientific honors include the Fritz London Award in Low Temperature Physics and election to the National Academy of Sciences. He chaired a 2009 National Academy of Sciences study on advanced radiation detectors commissioned by the Department of Homeland Security. He currently serves on the boards of Argonne National Lab and the Helmholtz Foundation in Germany, amongst others. Bob knows the world of tech transfer from the 35,000-foot level, and he also knows it on the ground. He's been a faculty client of our UC San Diego Tech Transfer Office for most of our 20-year history, with both issued patents and exciting new inventions in his portfolio. Please join me in welcoming Bob Dynes.
Thank you, Jane, and welcome all of you for this typical San Diego day. You're here learning about biotechnology and technology transfer, and the first person that talks to you is a physicist. I'm proud of that, actually. I'm really pleased to welcome you here, and I've had the opportunity for about 10 minutes to share some thoughts about the future of biomedical innovation. I'm delighted to precede the four speakers who are friends of mine. They're colleagues of mine. We have shared at least the last 20 years at UC San Diego, and I'm going to tell you, I'm going to come back to them in a minute, but I'm telling you that they're really, really creative, very smart guys, and they're personal friends. I want to follow up on Jane's introduction by telling you about a little bit about my own history, because it's relevant to what we're going to talk about and discuss this afternoon, and how I actually I moved my own ideas in technology. As a technology inventor, I moved from one coast, i.e. the East Coast, and one era to the other coast, the West Coast, and a totally different era, and I'm going to explain that to you. I started my scientific career in the late 60s at AT&T Bell Laboratories, at a time when the U.S. commerce achieved world dominance. The U.S. basically owned the world at that time. This is my own opinion. You can debate this if you want, but you'll lose. It was the era of R&D, research and development, and there were very large industrial laboratories, and I was in one of them at Bell Laboratories, which spun out an enormous amount of technological innovation. In some ways, we were too successful. And over time, as industry spread their new technology around the world, competition became global, and it became fierce. U.S. companies were no longer dominant, and they became global companies, and in my view, too large. Industry began to shrink their research and lower their sights towards short-term goals and quarterly statements rather than decadal goals. I realized in the 21st century, American universities will have to fill some of the void of that research that was done in industrial laboratories in the United States in the 60s and 70s and 80s. And the universities would have to take some of the responsibility for the stewardship of the nation's long-range vision and intellectual property. So I moved. I left a declining powerhouse, and it was declining. So those of you that remember the history, AT&T was falling off the edge of the table, and came to an emerging powerhouse, University of California, San Diego. And at the time, this was 1990 or so, the time it was completely liberating for me. At Bell Labs, we had pursued a single mission of research to contribute to the long-term health of private telecommunications company. And we were successful, as most of you know. At UCSD and in the public universities in the country, we pursue three interlocking missions: education, research, and public service to benefit the public in an array of ways and a wide variety of fields, much broader numbers of fields and interconnections of fields than I experienced at Bell Labs. And yet, as we entered the 21st century, we still had an R&D mindset. Research and development. It was all called R&D, and we would pursue it, demonstrate its value, and then you'd hand it off to somebody and hold your breath and hope it was successful. That was the mode. You'd pass it off to somebody else who was responsible for actually taking it to products that would benefit society. That R&D era ended in my mind on September the 11th, 2001. And I will never forget, and probably a lot of you will never forget, the sight of the World Trade Center buildings collapsing and watching first responders trapped in a buildings. I knew as a research scientist in the telecommunications field that we had developed the state-of-the-art wireless devices that could have kept the responders and the people in the buildings in touch with the dispatchers that were on the ground and tell them how much time they had left. But those devices never made it into the hands of the responders. I remember looking on television thinking, no, we know how to do that. But we didn't. And I'll be haunted forever by that observation. That single day, in my mind, caused a transition to a new era. We transitioned from R&D to RD&D, research, development, and delivery, and we could no longer afford the luxury of handing off those responsibilities to somebody else. We had to move discoveries from the bench to the public domain efficiently, effectively, and as quickly as possible. To do that, universities had to work more closely with companies, more closely, person to person, and also to the end users, whether they are first responders in a crisis or bedside healthcare professionals, saving lives and responding as quickly as you can. Companies that develop innovations need incentives for their work, of course. If they invest time and resources in this process, they should expect an opportunity, not a guarantee, an opportunity to get a return on their investment. Universities also need to show a return on their investment in research. This is especially true for the public universities. I know this. I've both had glorious times and I've been beaten up seriously as the UC president, the president of the University of California. But when legislators, the governor, taxpayers, insist on getting economic and societal value for what they invest in higher education. This new era of R&D has forced all of us in the world of science and technology to change not only how we work, but also what we think about our work. It's just a different era. We can no longer afford to operate in silos. I don't like to hear people anymore say, oh, I just do basic research. I'm afraid I'm pretty rude to those people now. We can't put up those walls. Listening to people say, oh, those industry people, or oh, those academics. We're in the same room now. In this new era, generating new knowledge that will serve the public and benefit society is a team sport, full body contact. And no one knows better than technology transfer professionals than Jane and her staff here at UC San Diego, whom I've been working with. In fact, we just submitted a couple of patent proposals, and we're working with small companies. I've watched with great pride as that unit has grown over the last 20 years. Our four speakers today, Larry Goldstein, Shu Chien, Larry Smarr, and Nick Spitzer, are also longtime tech transfer clients. Each of them is a giant in its field. Together they constitute an all-star team that exemplifies what I'm very proud of at UC San Diego. They're world leaders in technological innovation. Scientists, but world leaders in technological innovation. Let me end with a few words about something the four of them have in common, and something that I hope we all have in common, and something that you have to remember all the time. When you listen to their presentations today, you'll undoubtedly think, geez, these guys are really smart. Brilliant, ingenious, but above all else, you will sense their passion for the knowledge and how they're using that knowledge. As a physics professor who teaches graduate students, I can tell you that the young scientists that I deal with, those with the greatest potential, aren't necessarily the ones who have the highest measured IQ, whatever that is, and the most impressive publications. In every generation and in every field, the people who feel the greatest passion for their work will have the biggest dreams and take the wildest risks. Passion is what drives innovation. And that's where they're successful, and that's why we're here today. Our succession moderator has brought passion to work as an intellectual property attorney and has fought the good fight for startup companies and universities in this region. He has an outstanding track record in patent and trademark protection, but he is, at heart, a scientist with a degree in electrical engineering. Not quite a physicist, but almost. But he's dealt with patents that have been licensed, enforced, and sold. Please join me in welcoming Steve Fallon from our symposium's gold sponsor, Greer, Burns, and Crain for a few words. Thank you.
Thanks very much, and yes, it was electromagnetics that convinced me that I was not a physicist and sent me to law school. I do share the passion for innovation, especially at the high level, and to sit among our speakers today is a great pleasure. One of the advantages of working with high-level RD&D institutions now is to see the best of the best, truly all-stars. Not only all-stars in their research efforts, but all-stars in their communication efforts, their communication with private industry, the communication with attorneys, the communication with private companies as well. Jane had mentioned that today was a happy, this week is a happy coincidence because the bioconvention is here and many of us will be attending the bioconvention. It's also a happy coincidence that this is on the 20th anniversary of the Technology Transfer Office here at San Diego. A happy coincidence is that it is also my firm's 20th anniversary, and I've had the great pleasure of working with the TTO for most of my 20 years at Greer, Burns & Crain. The TTO is a highly professional agency. I know firsthand of their interactions with faculty, with outside counsel, and also with private industry. Hundreds of startups have resulted from the TTO's efforts. The TTO has licensed technologies throughout, certainly in California and throughout the United States, in Europe, in Asia, and even in Australia. They're widely recognized for their professionalism and for their integrity in every encounter that I'm aware of. Our first speaker today, Dr. Lars Goldstein, is celebrating his 21st anniversary here at the university, and pardon me for mentioning 21, and one of the interesting things that I discovered about Dr. Goldstein's research is it's not just advocacy in the sciences, but it's advocacy in the political arena, it's advocacy in the public arena, to engage the public and help the public understand the importance of the research that is going on at institutions. Dr. Goldstein has testified before the Senate in the days of controversy regarding stem cell research in 1999. Dr. Goldstein went before the Senate committee and explained the importance of maintaining funding in public institutions as well as private institutions, recognizing the talent that exists in our public institutions here. For my benefit, Dr. Goldstein is also an author of the Stem Cells for Dummies book. And reading a review of the book, it was well received. It was not determined to be overly ambitious, although all of our all-star researchers are certainly overly ambitious. And it was reviewed to be an excellent guide for someone having an answer to, or needing an answer to a specific question. This research is sure to lead towards innovations that will help many treatments that are personal to so many people, from Alzheimer's and other degenerative diseases. And it is with great pleasure that I introduce Dr. Goldstein today.
It is amusing that this is something of an anniversary. 21 years is interesting. So I guess I'm old enough to drink at the University of California now, finally. But the other thing that struck me as I was listening to Bob Dynes is that when I first moved to UCSD from Harvard University, it was also an east to west odyssey. But interestingly, I first knew Bob as a scientific colleague when I arrived at UCSD before he became an administrator, but we think you're still part of the team anyway. That's good. And it was meeting him as a physicist that was actually very important to me, because as a newly arrived molecular biologist at UCSD, not only did I already have built-in interactions with folks like Nick Spitzer, who was a well-known neuroscientist at the time, but very soon after my arrival, I was interacting with physicists and engineers and physical chemists and people in other disciplines, and it led to a great many collaborations. Some of them led to the formation of a company that I still hold dear, and it's part of what makes this institution, UC San Diego, and the surrounding institutions so incredibly interesting to work at, is the degree of intellectual interactions outside one's discipline in a very freewheeling sort of way that just didn't exist on the East Coast, and doesn't exist at most institutions, to be honest. And so what I'll do, let's see if I'm going to get this right. How does this work here? Oh, there we go. Okay, good. All right, so that's me. And so what I'll talk about is stem cell science. And what I'll give you is sort of a three-part story. The first part is just to remind you of the basic background of how stem cell science works. It'll be a little more advanced than stem cells for dummies, so I will not go back to full lay language. You guys are far more sophisticated than that. But I do want to make sure that we define our terms and agree on what we're talking about. I'll be returning to Bob Dynes during that part of the presentation, as you'll see, so we'll keep him on the edge of his seat. And then I'll talk a little bit about how my own lab has been using stem cell technology to probe and to develop therapies for really terrible diseases, Alzheimer's disease and Lou Gehrig's disease, ALS. And then I'll close at the end telling you how we as an institution at UCSD are riding the next wave of stem cell science into the development of useful therapies as a result of the formation of a new program at UCSD with the help of our colleague and friend, Denny Sanford. So I have to start with the usual financial disclosures. You folks are professionals. You know how to deal with this kind of information. I'm subject to personal financial gain. I have biases because of trials and other activities at the university. My friend Irv Weissman sometimes says things like, well, from here on out, you shouldn't believe anything I say because I'm so biased by my conflicts of interest that I couldn't possibly be objective. So I'll leave that to you to decide. And the background for this really does start about 15 years ago in the U.S. Senate, because up until the announcement by Jamie Thompson's group in Wisconsin of human embryonic stem cells, I had no idea that I would be working on humans at any time in the near future. And the reason I was asked to testify in this hearing in the Senate, in a subcommittee chaired by then Senator Arnold Specter, was because my lab and I had worked on mouse embryonic stem cells for a number of years, and therefore I knew something about the technology and what one might do with it in the human. In the first hearing that I testified in, one of the folks I testified with was Christopher Reeve, then an actor, and as many of you know, also a victim of a horseback riding accident. Christopher had a break in the cervical region of his spinal cord, and as a result was basically paralyzed from the neck down with very, very minimal function. Chris dedicated the rest of his life to the support of research to develop stem cells and any other potential technologies into useful therapies for spinal cord injury, which is a very common injury in our society. But Chris also had a way of capturing what I would call inspirational statements in very plain language. And an example of that is shown here, where he talks talked about what really drove him was the notion that medical research and then translation, R&D, as Bob puts it, really had the potential to take words like impossible and unsolvable, which was the state of spinal cord injury therapy 15 years ago, to the realm of actually changing the ability to treat and actually reverse and eliminate some of the symptoms as a result of these injuries. And as you'll see at the close, while we're a few years too late for Christopher, we are about to launch spinal cord injury stem cell trials at UCSD. Other institutions and companies around the world have already begun, and actually it's turning into a bit of a hot competition to see which of these therapies will work and then work best for the treatment of this sort of injury. It's really quite remarkable what's happening. Now, let's start at the beginning, and that is with the question of what are stem cells? What do I mean when I say stem cell? There's a lot of misinterpretation of the term in the public and business domain. And then what do we think about how they can help? I've got to keep an eye on the clock here. Okay, good. And so let me give you a quick definition. So if we go back to Bob, this is the part where Bob is the audience participation. If we go back to Bob, and should I make him stand up? Yeah, what the heck? Okay, Bob. So we're going to stand up. Oh, come on. Yeah, you. So Bob's going to stand up, and we're going to look at Bob, okay? So we're going to make him the center of our attention. Not a pretty sight. Not a pretty sight. No, actually, he's a very handsome gentleman, aging very gracefully. But that's not the point I'm going to make. So what you'll see is that, you know, we learned in high school biology that cells grow by making more cells by cell division. And we learned that those two daughter cells from a cell division were identical, right? Okay, that's what I call the biggest lie in biology, and you guys already know why. See, if we look at Bob, Bob is not just an accumulation of identical cells, right? He's got hair cells, eye cells, skin cells, muscle cells, plenty of those, heart, lung, liver. Okay, I'm done with you for the moment. So he's the demonstration of why that basic dogma can't possibly be correct in its simplest form. And part of the resolution of that dogma, not all of it, but part, is that there are special cells in our bodies that during development, when they divide, the two daughter cells are not identical. That's important, because one of the cells remains what we call a stem cell, and the other cell, if it gets the right kinds of signals, can go off and become a hair cell or a heart cell or a liver cell in a controlled way. So ultimately, a stem cell is a cell that we define as one that can divide to make more of itself, as well as its progeny responding to biochemical signals to make specialized types of cells, such as those we find in an aging, gracefully gentleman, such as Bob and myself, I'm afraid to admit. So that's actually important, because that means, if you think about it, that this is a very important enabling technology in biology and biomedicine. And so I'll return to a second example, which is a film that was made in the 60s with Dustin Hoffman as a character who was trying to figure out what his career path was going to be. And he received some very important advice at a party he attended just after he graduated from college. I'm sure you folks all remember the career advice Dustin Hoffman received. Plastics, there you go, that's your cue. Okay, very good, you're paying attention. And in the 60s, that was the right career advice, because plastics were the future of manufacturing in many ways. We were no longer going to use steel and wood for everything we built, we were going to use plastics. And the result of that you can see in this building, in this room. Many things we used to build out of natural materials, we now build out of plastics. It was great career advice. Well, in 2014, the advice I would give is biological plastics. And what I mean by that are stem cells, because stem cells give us the ability to control the development of different sorts of biological materials, to develop cells that might replace those lost in disease, or to give us a way of studying human biology in the lab using human stem cells that we were never able to do in the past. And so the coming era is one of biological plastics, where we will learn how to use these cells to tackle problems in disease, to develop new bits and pieces of organs, and I suspect to do things that none of us is smart enough to anticipate. That's the beauty of enabling technology. When smart people come into contact with enabling technology, they think of things to do with it that you never would have imagined, right? Look at recombinant DNA and its use in forensics. You never would have predicted that in the 70s, right? So it's a totally amazing thing that happens when useful enabling technology gets into the hands of largely smart people at public and private institutions. And so stem cell technology, as you'll see, is giving us a way to tackle problems that have, as Chris Reeve would have said, been previously intractable. And in the case of Alzheimer's disease, ALS, and spinal cord injury, I'll give you examples of how we're doing that now. Now, one of the confusions in the world is that, first of all, there are actually many different sources of stem cells. You can get them out of cord blood, you can get them out of amniotic fluid, there are so-called embryonic stem cells, and there are now reprogrammed stem cells, a technology that won Shinya Yamanaka the Nobel Prize a couple of years ago. And the message I'll just leave you with as tech transfer and business development professionals is that when somebody says stem cell, the first question you should ask is what kind? Because just as all of the different tools in a toolbox have different uses and different sensible uses, you wouldn't use a screwdriver to hammer a nail, for example, although that's a creative use of the technology, I suppose. In fact, knowing what the different types of cells are when somebody approaches you with some great idea will help you understand how best to develop it and think about it. I'll note, for example, that there are clinics all over the county and the world that will propose to take stem cells from your fat and use them to treat every disease known to mankind. This is actually an example of fraud, it turns out, because the cells that they claim to be generating, so-called mesenchymal stem cells, we now know as a result of scientific presentations I heard in the past few days at the International Society for Stem Cell Research meeting. The claim is that these are so-called mesenchymal stem cells, and the fact is whatever these folks are getting out of fat is not. But if you look, for example, in bone marrow, you can find cells that legitimately make all of the cells of the blood system and the immune system. Embryonic stem cells, which come from early frozen embryos left over after IVF treatment, those cells can be grown to quite large quantities and can make any cell type that we find in an adult human, such as Bob or myself. And reprogrammed stem cells have many of those same kinds of properties and give rise to all sorts of different kinds of uses, as I'll illustrate now. And so the second part of what I want to talk about is how we and others are taking this kind of stem cell technology, where we can grow large quantities of cells and direct them to generate different sorts of adult cells, and how we're using them to tackle, in my case in particular, problems in neurodegenerative disease such as Alzheimer's, Parkinson's, ALS, and others. And of course, the background, as you know, is that we currently have no drugs, no useful therapies that change the course of any of these disorders. We have drugs that help with some of the symptoms, but there is an incredible and pressing need for drugs that will slow the development of these diseases in a person who has them. It's a terrible need, actually. And so one of the approaches that we and others have used that takes advantage of stem cell technology is so-called disease-in-a-dish technology to tackle problems such as Alzheimer's disease or others. And the basic notion is, if we return to Bob, if I took a sample of Bob's skin cells with a small biopsy, I could then generate so-called reprogrammed stem cells that have Bob's unique genome, his unique DNA, in stem cells that I could then use to make neurons, brain cells, other types of brain cells called astrocytes, and I could study how those cells interacted in a dish and use them to test for drugs that would perhaps reverse Bob's personal set of disease and disorders. And I'll come back to that in a moment. The particular case that we've been using this for that's illustrative of this technology is the case of Alzheimer's disease, where we know a great deal about the development of pathology and the loss of connections between brain cells before they die but where we don't really know what the basis biochemically and cellularly is for both the formation of the pathologies in most people who develop disease, and then in particular, how some of the detailed biochemistry of this protein that I'm pointing to right there, called amyloid precursor protein, which we know is part of the development. Can you see those? Oh, I can see this little thing and you can't. Well, too bad. Down at the bottom there, we know that protein is integral to the development of pathology and disease, but we don't know exactly how. And so one of the things we've done in my lab, and this is work from a really smart graduate student of mine, Mason Israel, was to take a number of individuals who either had rare hereditary forms of Alzheimer's disease or who had what we call sporadic Alzheimer's disease. You guys know that sporadic disease is code for we don't know what the heck caused it. It's the kind of disease most of us will develop. And then some patients from our Alzheimer's Center who are controls. And we reprogrammed them using this technology and used them to develop, as shown here, purified neurons in a dish that either had the genetic changes that caused hereditary Alzheimer's disease or the genomes, the DNA of people who developed sporadic disease, where we know there's a big hereditary contribution, but we don't know how or why, and also controls. And then compared the properties of those neurons carrying all these different genomes biochemically in the lab. And what Mason learned was that we could recapitulate in a dish, amazingly, some of the abnormal biochemistry that we see in early stages of Alzheimer's disease. And that's what this slide illustrates. And without a pointer, it's going to be a little hard for me to point to it. But if you just look at the far right graph there, you'll see that the two rightmost bars in that graph are high. The two leftmost bars are low. That's controls compared to cells from people, cells with the genomes of people who had hereditary disease. And you can see that there's a big difference in one of the downstream biochemical changes that we see in these cells and in the development of this disease. And what's remarkable about this ability to both generate cells in a dish that carry the DNA of people with particular forms of disease are that we can see these very simple early biochemical changes, but that using other genetic tricks, we can elucidate what the biochemical steps are in the lab in these pathways. That's a really interesting problem, it turns out. But even more important, using this very simple kind of assay, we've started testing FDA-approved drugs with folks at the Sanford Burnham Medical Institute in their Prebys Chemical Genomics Center. We're looking for FDA-approved drugs that change these biochemical features of disease in a dish. And remarkably, we have already, even in a few months of work, it took a while to get this to work properly, don't underestimate the difficulty here, but once we've gotten this to work properly, we have found a number of really interesting hits that you wouldn't have found by drug testing by any of the means that pharmaceutical industries conventionally use. And so we think that these can very quickly, after we finish characterizing them and figuring out what their mechanisms are, get them into early stage clinical trials to begin testing, will these really be efficacious for actual disease in people, leapfrogging off this very relatively simple cellular set of assays that we can pull off in the lab. So that's one example of so-called, oh, thank you, Nick. Wow. Yes, that's the graph right there. You just remembered you had the pointer? Somebody gave it to you as a Christmas present, whatever. Thank you. So it's a remarkable technology, and that's one example of it. Now, the second way this technology is turning out to be incredibly useful is to dissect how human genetic variation predisposes to common, sporadic forms of disease. And that is this ability to capture in stem cells the genomes of individual humans. And that's important because if I want to study neurons, brain cells, that have the DNA of individual people, if we return to Bob again, if I were to say, Bob, you want to come up to the lab this afternoon, we'll get the drill out, we'll drill a little hole, scoop out some of your brain, Bob's response to this would be, Hell no. Hell no, that's right. I've gotten that response a lot. I did get one yes once. That person should have their head examined, and we were about to do that. That's right. So we can't get Bob's neurons in a dish without doing this method of taking skin cells, reprogramming them, and then we can generate Bob's brain cells in a dish. We could actually generate Nick's brain cells in a dish. We could generate a mixture and see how they talk to each other. This technology is incredible for studying cell interactions. And so we've recently done that with Craig Venter. So Craig, you know, completely sequenced his own genome with very high accuracy and fidelity, and so as a result, he's one of the rare genomes where we know all of his risk factors for the development of disease. And so Craig let us biopsy him, and then we used the so-called Magic 4 retroviruses to reprogram his cells into stem cells, and folks in my lab have now literally been studying Craig's brain cells in a dish. And not only are they aggressive and megalomaniac, but they actually have things to teach us about the variants that predispose him to disease. And in fact, we've now sampled a large number of individuals who have different variants in their genomes, and we've discovered recently at least how one of the genetic variants that is known to predispose to disease causes biochemical changes in brain cells that have that variant. It's taught us a great deal about how we might drug that pathway, and it's also told us how to go about stratifying clinical trials potentially so that people who have different variants, we would actually make different predictions about how they'll respond to certain kinds of drugs. And we just wouldn't have gotten that kind of insight without this ability to probe how variation predisposes in a dish. So the final example is ALS, Lou Gehrig's disease, where we know that in the development of this disease, what happens is that people progressively lose their motor neurons that control movement of their muscles, and ultimately, after all of those motor neurons have died, these people are completely paralyzed. They can't breathe, they can't swallow. It's really terrible. Some of them go on ventilators, feeding tubes, what have you, but it's a very devastating disease. Usually, average lifespan after diagnosis is 3 to 5 years, and there is no drug that really changes that basic timeline very much, and some people die within a year. It's a terrible, terrible disorder. So the question is how to go about using this kind of technology to treat Lou Gehrig's disease successfully. And there are a number of labs around the US and the world that are using the so-called disease in a dish approach to try to devise drugs to treat Lou Gehrig's disease. We've taken a slightly different approach because as a result of work we've done in the past decade with my colleague Don Cleveland at UCSD, we know that even though it is these long motor neurons that are dying, other cells in the spinal cord called astrocytes play a very important role in potentially speeding the death of motor neurons or slowing the death of motor neurons if we manipulate the astrocytes directly. And so that has led to a project that I currently lead, where what we're trying to do is to start with human embryonic stem cells, develop in the lab by taking advantage of these cells' ability to differentiate and grow in vitro. We generate precursors to neurons and glia, astrocytes, in a dish, and we know that if we transplant these into experimental animals, not yet the human, but soon, if you're a rat or a pig, these cells will differentiate into large numbers of functional astrocytes in the spinal cord of these transplanted animals. And so the hope is that we can actually get these cells into clinical trials in the next few years in ALS patients to test whether we can slow or reverse the course of that disease. And there are other groups out there using different types of neuronal precursor cells, and the point is it's really only through clinical trials that we'll be able to understand which types of cells will slow or, we hope, at some point reverse the course of this disease. Now, as Bob mentioned, and this is the final part that I'll hit briefly, is to talk about how we and others are taking this kind of science and using it to develop appropriate and rapid clinical development programs at the University of California and elsewhere. Bob mentioned that guys like me who spent our lives at other institutions prior to moving to UCSD, such as Harvard, learned that most of the academic world is built as silos, so that you're in a department, for example, the Department of Corn, and if you wanted to talk to somebody in the Department of Wheat or soybeans, you were pretty much out of luck. But of course, what I told you at the beginning is already when I arrived as a biologist, I was quickly speaking and interacting with really smart physicists such as Bob, engineers such as Shu Chien, and a great deal of other individuals. And of course, that's the point of how we do things in San Diego, and it's what led to the development of the Sanford Consortium for Regenerative Medicine, which in a sense is a set of bridges between institutions and disciplines in the area of stem cell research with the goal of developing good ideas that might then be translated into clinical medicine. And of course, the next step in this progression, as you may have recently heard, is that, again, our friend Denny Sanford stepped in with a large gift to UCSD to establish the so-called Sanford Stem Cell Clinical Center, which is really dedicated to accelerating stem cell-based clinical trials in humans. It really is the only way, as you know, to develop effective treatments is at some point you have to do research with human subjects. And so the major goal of the center is to combine ethics, education, and a hell of a lot of collaboration in science and medicine to bring new therapies to patients over the next decade or so. It's a 10-year and more project. My job is to make it a sustainable project at the end of 10 years. I have a lot of work coming up. And one of the basic means of doing this is, as you guys know, one of the biggest obstacles to development of good scientific ideas and discoveries into useful therapies, we have to go through the so-called valley of death, where there really is a dearth of risk capital in the world and a variety of other problems. And so part of the goal of the Sanford Stem Cell Clinical Center, if you will, is to live in the valley of death, and in a sense to function, as you'll see here, as a big funnel. And you're going to ask, what the heck do you mean by a big funnel? And really the idea is here, and that is to find ways to take and accelerate scientific discoveries from our partner institutions in San Diego and elsewhere as needed, and to bring them as rapidly as possible to experimental trials and hopefully the development of treatments by taking advantage of this unique gift from Mr. Sanford. One example of that is actually shown here, where because of Mr. Sanford's gift, we have been able to accelerate the development of a phase 1 clinical trial for spinal cord injury using cells that come from a company called Neuroscience. These are fetal stem cells that will make neurons and astrocytes and other types of glia following transplantation. And what's remarkable is shown at the bottom, which is if you're a rat, we don't yet know about humans, but if you are a rat with a spinal cord injury and these cells get transplanted to the site of injury, they will grow in a way that adult cells just don't seem to. Long fibrous connections in both directions across the injury site in the injured rat. And these cells appear to establish contacts with cells both above and below the site of the injury. And again, if you're a rat, there is large restoration, though not complete cure, of function. And so we cannot continue experimenting on rats forever, and as a result, we will begin clinical research with humans with these cells. We expect to do 8 patients in the next 6 to 8 months to see how humans respond to this kind of transplantation. We are hopeful, but as you know, clinical research in humans, we are not just big rats, you may have noticed, and so the response to the cells may be different, but it's a really important next step. So let me close there. I've used up my time. I hope that you've gotten a sense of the kind of excitement that is happening around here in San Diego in these fields, and maybe to get a taste of how we intend to accelerate their application in human patients. Thank you. Okay, so what happens now? Do I answer questions, or do I give up the microphone? Or
does anyone have any questions for Dr. Goldstein?
You may have read my book, so then there'll be nothing out there to tell. Yeah.
So as your work progresses with Alzheimer's, will the results be translated to other diseases?
So the question is, as we make progress on Alzheimer's disease, will the results be applicable to other diseases? And I think the answer is yes. So all neurodegenerative diseases share a couple of features, it turns out. One of them is the early loss of connections between neurons, and the other is some of the pathologies that develop overlap greatly between different diseases. And in fact, some of us joke that the different diseases are categorized with names because the neurologists need to know which branch of the hospital to refer them to. And in fact, we know in some cases that similar genetic defects can give rise to either autism, schizophrenia, or bipolar disease, depending on which individual has them. And we know there's a great deal of overlap, for example, between Alzheimer's and Parkinson's. So we suspect that there will ultimately be a great deal of so-called off-label use, and that the field may evolve in the way that cancer has evolved, where you're not categorized necessarily by which part of your body has cancer, you know, prostate or breast or liver or what have you, but you'll be categorized by the genetic change that has driven the development of aggressively dividing and migrating cells. And that's a very important realization being capitalized on now at the Moores Cancer Center, where, as some of you may have heard, a company of Craig Venters that I freely disclose, serve on the board of, they're going to sequence every cancer patient who walks through the door to begin understanding how their genomes predispose. And so I think it's a really interesting set of projects, but I think this will be similar in the neurodegeneratives as well. Yeah.
So Larry, so we've got in San Diego now world-class research and development on stem cells. We've now got world-class clinical. The other part of that equation, the kind of the third leg, is the manufacturing, the ability to really manufacture these in a GMP fashion. How do you see us getting into that area? It's a loaded question.
It is a very loaded question, Paul. So as Paul knows, there is an ongoing study in San Diego to ask whether we should construct a manufacturing center here in San Diego for the transition of stem cell therapies from what will be small phase 1 trials, where one doesn't need to manufacture large, huge quantities, to phase 2 and phase 3 trials, where you potentially need to treat hundreds or thousands of individuals. And so whether or not we should build one here in San Diego is a great question. It's an ongoing debate. Certainly the governor of Texas has thrown down the gauntlet as to whether he thinks it should be in Texas or not. I think we should never let Texas get a leg up on us here in California and San Diego, but we have to make sure that if we use civic support to build something, that there is adequate levels of need to support and sustain an operation like this in the coming years. So it's a really hard kind of problem, but you know, you study it and then we'll come up with some sort of recommendation. Great, thanks.
Special thanks to Dr. Goldstein. Our next distinguished researcher is Dr. Nick Spitzer. Dr. Spitzer, I hope you won't mind me mentioning your anniversary here at UCSD. Dr. Spitzer has been at UCSD for, on my count, 41 years now. So you've reached the drinking age twice here almost. Dr. Spitzer, appropriately, Dr. Goldstein laid the groundwork for us, and Dr. Spitzer will talk to us now about the future of research in neuroscience. And please join us in welcoming Dr. Spitzer to the stage.
At this point, I had the opportunity to be drunk several times over, so hopefully I'll stay sober for the presentation this afternoon. I'm very glad to be here to talk about the future of neuroscience broadly and what it means for us here in San Diego. The future of neuroscience is unfolding very rapidly, much like that Larry has described for us with stem cells. A big part of it will be in neurotechnology. And I'm going to try to focus in on this. I think it's going to be a technology that will be, in many ways, complementary to the technology that Larry's talked about. And as our last question brought up here, at the end of the day, this is going to be about companies, translation, patients, clinical trials. I think this is going to be happening here in San Diego. So this afternoon, I plan to touch on three topics in my remarks. The first will be to just sketch briefly for you some of the very exciting results that have emerged in neuroscience research, actually just during the recent few years. Then I'll talk at some length about the BRAIN initiative that I'm sure you've all heard about, the federal initiative, President Obama, and how San Diego was critically involved in actually promoting and launching this big presidential grand challenge. And I'll mention briefly how that's going to unfold nationally, but at the end I want to focus on what it is going to mean here at San Diego, what we're doing, and where we think this will take us. So when we think about the brain, it's a human brain here, we are immediately aware it's a very complex structure. Chatting with Bob earlier, he was quick to point out, even as a physicist, that this is arguably the most complex structure in the known universe. And we have about 100 billion neurons, many more of those glia, including the astrocytes that Larry told us about. And each of those neurons has about 10,000 inputs. We call those synapses. These are the connections that Larry referred to. And so in this cartoon here, this very simple cartoon of the brain, we have processes like this axon coming down, making contact here with this dendrite, and this is, of course, the site of these synapses. So there are trillions, literally trillions of these synapses in each of your brains, and also in Larry's and in Bob's. So a lot of interest focuses on these synapses. They contain neurotransmitter molecules. This is on the so-called presynaptic side. They contain receptors for these molecules, because these transmitter molecules are released when an electrical signal comes flashing down the axon. And we're interested in these for two reasons, fundamentally. We talk a lot about plasticity, a topic that Larry's introduced for us this afternoon, initially with the wonderful remembrance of plastics as the appropriate career choice. Plasticity in this context means changes in the brain in response to changes in the environment or to experience. We would put in here, of course, learning and memory. This is one of the kinds of plasticity that we always want to keep fostering even after many years at UC San Diego. A second reason, then, in addition to the fact that the learning and memory changes occur here at the synapse, is that the neurological disorders that, again, Larry has alerted us to here, many of these disorders, not only Alzheimer's, but also schizophrenia, depression, disorders of this kind, are thought to result from malfunction at the synapses. So let me just tell you briefly about a couple of recent studies that have been going on at UCSD that highlight the very high quality and the impact of the work that's being done there. So here's a story, a recent story. This came out just earlier this month in this magazine, Nature. You know this magazine. This is a well-known magazine. And this is the work of Takaki Komiyama, a brilliant assistant professor on the campus now, only been there a couple of years. And it's looking at motor learning. So what's motor learning? Motor learning is what we do when we learn how to swing a golf club or to hit a tennis ball or even to ride a bicycle. Initially, when we start out, we make a lot of mistakes. And the trajectory of our swing is very uneven, and it's very variable. But over time, maybe quickly, we become very adept at these motor behaviors. And our movements become highly stereotyped. And what Takaki and his colleagues have done here is to take a mouse, be able to peel away the skull, image hundreds of neurons in the motor cortex of the brain over a period of weeks when the animal is learning to perform a motor behavior. In this case, it's licking to get a drop of water. Mice are thirsty, just as you. And as they start to do this, the firing of the neurons very heterogeneous, but over the course of just a short period of time, the neurons begin certain constellations of neurons begin firing in synchrony, and this is a result of the coordinated activity through synapses in the brain. So a fabulous physical insight about how many neurons are involved in this learning process, how they start to get together. In another study, again, very recent, in fact, it's so recent, published in Nature, that the citation has not yet appeared. This is only available on the internet. This is the work of Roberto Marano, a National Academy member there at UCSD, along with another Academy member, Roger Chien, a household name for us, a Nobel laureate. And they've been able to engineer a memory with long-term depression, LTD, and long-term potentiation. So how do memories occur? How are they actually produced? We've thought for a long time that it's a result of high-frequency activity at these synapses that changes the property of the synapses and produces this long-term potentiation. What they've actually shown now, and again, this is in mice, but presumably this is the way it's working in our brains, is that they could generate a memory. It was the memory of a tone that the mouse would hear, and in response to a foot shock, it would demonstrate a particular behavior. They could then abolish that memory by a particular kind of stimulation to produce long-term depression, and then they could reinstate the memory with long-term potentiation. This was headlines wherever you're from. You may have seen this. This was a really dramatic demonstration of the physical substrate of memory in the brain. A remarkable accomplishment up here at San Diego. And finally, just briefly comment on some work that we've done looking at transmitter switching in the adult brain and how that regulates behavior. This was fun to do. This was a result of a lot of work over a sustained period of time, but with my colleagues, in particular Stefan Leutke, recently promoted, now associate professor at UCSD, last year, we showed that in a rat, presumably the same thing will happen in a human brain, in response to different sensory stimulus. In this case, it was day length, photo period, long day, short day. Right now, we're enjoying the longest day of the year here in San Diego as elsewhere. This can change the identity of the transmitters in particular regions in the brain. This was a scientific heresy. This was thought not to happen. We thought that transmitters were fixed, unchanging, constant, invariant throughout the life of the organism. Turns out, not true. And remarkably, when that transmitter switches, so does the behavior of the animal. So I hope you're getting a sense here that there's a lot of very exciting neuroscience going on at UC San Diego in these areas, in addition to the areas about which Larry told us. So, how did the Brain Initiative get launched? This is a wonderful story, and it's very much a UC San Diego story. This began in 2011, so not that long ago, with a meeting in England at Chichley Hall at the Kavli Royal Society International Center. This is a wonderful English estate, a fabulous place, and it was the brainchild of the Kavli Foundation, thinking that it would be very important to bring together engineers, nanoscientists with neuroscientists and see what kind of cross-fertilization, what kind of interaction could arise in this kind of environment. They met for 3 days, they talked, they discussed, and they realized that these two silos, again coming back to Larry's metaphor here, actually had a lot in common, and that corn could talk to wheat, and that there was going to be an opportunity here to make things happen. Within a month, they had developed a white paper, the whole brain activity map, merging nanoscience and neuroscience for technology and health, and delivered it to the OSTP, the Office of Science and Technology Policy. So this was moving pretty quickly. Enthusiasm built very quickly over time, and by June of the following year, the Brain Activity Map project was now identified. The idea was to understand at a functional level how nerve cells are connected to one another. We're beginning to get a sense of how that works at the anatomical level. Of course, we want to understand how that works at a functional level. And this enthusiasm continued to build. Brain Activity Map, a nice report in Science in March of 2013. But even before this article appeared, talking about how one could make progress on this front, we were blown away in the State of the Union address in February of 2013 when Barack Obama, out of a clear blue sky, of course not quite a clear blue sky, here with Joe Biden and John Boehner, talked about the challenge of mapping the human brain. And so this was something that started with Ralph Greenspan, one of our faculty at UC San Diego, Terry Sejnowski at the Salk Institute. These two gentlemen were on the ground at the very beginning. I had the good fortune to join them shortly afterward and respond then to this federal challenge, this grand challenge of moving ahead to map the human brain. In April, so this is February, now in April, there were a group of us, Pradeep Khosla, our chancellor, Ralph, Terry, myself, and others. We were in the White House in the East Wing, and we were there to hear in more detail about the BRAIN Initiative. So here's Barack Obama. We all recognize him. Many of you will know Francis Collins here on the left, the director of the National Institutes of Health, and he's introducing the man whom he characterized and who was very proud to be the neuroscientist in chief. And so this was terrific. A lot of enthusiasm, a lot of public press on this point. And for us here in San Diego, it was terrific, as you'll see, because it led to something that's very tangible, very concrete, that I think will have lots of legs for us here in town. So what was the BRAIN Initiative? What is the BRAIN Initiative? In broad terms, it's on the order of about $100 million for fiscal 2014. And this is with the objective here of doing some very good, very useful things to begin to understand how the brain actually works. It's phenomenal that after really a century of hard work on this problem, we are remarkably ignorant about that process. Many of you will be thinking, as we did too, this is actually small money because the support through the National Institutes of Health alone for neuroscience is on the order of $5 billion. That's with a B annually. So this is really a tiny fraction, but it's in many ways comparable to the 01 year of funding that was provided for the Human Genome Project. The challenge, of course, is figuring out how are we going to spend more money. One needs to begin to sort of seed the ground, get things started, find out where the money is going to be usefully placed, how much money can we give to Bob, how much money can we give to Larry, and so on. So this looked like a very good start, and it's going to be distributed in the following forms. The Defense Advanced Research Projects Agency, DARPA, that many of you will be familiar with, is going to be the high roller here, coming in with about half of that amount. National Institutes of Health going to provide about $40 million, and then the National Science Foundation is coming in at the end here with $20 million, so a little over $100 million, but a very good way to start the process here. And then there's going to be substantial support in this initial period, and then continuing forward from a number of private supporters. So the Allen Brain Institute in Seattle is going to come in at $60 million a year for the next period. The Howard Hughes Medical Institute, HHMI, is going to make a very substantial contribution. Kavli Foundation is going to contribute in proportion to its ability to do so, and the Salk Institute has ponied up at a very nice level here to provide support for this BRAIN initiative. Importantly, and Larry referred to this in his remarks, it's very important in these large efforts to focus also on some of the ethical issues that arise here when we come to work on the stem cells on the one hand or the brain on the other hand. These are important issues that go to the core of the health and the identity of individuals, and so it's important to consider that very thoughtfully. But I hope you're noticing what's at the bottom of the slide here, because this, in our minds, is almost certainly the major driver in Barack Obama's mind. And this is the economics of this BRAIN initiative. Let's just remember what the story was for the Human Genome Project. This was a $3.8, roughly $4 billion investment over a period of time. And the remarkable thing that came about, and this was the result, this was analyzed, the results were analyzed by the Battelle Institute. They studied the economic impact of each dollar that was invested in the Human Genome Project. And it turned turned out there was an enormous multiplier, I think about $141 of economic return for every $1 invested. So this is very impressive historically. It's been great, of course, for the country, and I'm pretty sure that Barack Obama had this in his mind when he thought about the BRAIN Initiative, and I'll come back to that a little later. So, how to spend this money? The National Institutes of Health went about this in a very thoughtful way, and I want to highlight this for you in a very particular way. They put together a blue ribbon committee that over the course of the last year now, since April of 2013, has been studying and thinking and meeting and consulting with experts on how to best deploy this money, what kinds of questions should be the number one questions, what questions are the number 2, 3, 4, and questions. And they came up very recently now, June 5th, with their report. It's the composition of this committee that I want to highlight for you. Here are the 15 individuals, and I've framed here in light green the 6 of the 15 that are from the state of California. So California has a very big stake in this process. 3 of these individuals are from Stanford, 1 from Caltech, and 2 from San Diego. And we've already mentioned them, Roger Chen and Terry Sosnowski. So California has a big stake in this process and is already influencing the direction of this spending of this money, and I think it's going to turn out to be a very useful thing for San Diego. So let's think for a moment about the large initiatives that we've had in this country, national initiatives. All of you, I think, will remember, perhaps many of you were around at the time, the declaration by John F. Kennedy in 1961, we're going to put a man on the moon. This was a visionary challenge. It was a very ambitious challenge at the time, and nobody knew how to do that. But of course, it delivered the goods. A little later on, a decade later, we had the war on cancer. And that was harder to achieve. I think the war was fought, as you heard from Larry. It continues to be fought. It's not a war that we're going to win anytime really soon, but we're making good progress on that. And importantly, the efforts of the war on cancer catalyzed an enormous amount of progress, tool development in the biological sciences, and that I think should be counted as a real win in the war on cancer. More recently, as I've mentioned, we had the Human Genome Project, 15-year project, and again, we've been able to sequence some genomes, and more of that is happening, as you know and as we heard. But again, much of that success of that period was in developing new technology that has been very helpful to us. So finally, here we are with the 2013 BRAIN Initiative. What is it about this initiative that is likely to lead it to be successful at the very high level that we anticipate and want from a national challenge? Well, I think that the first thing to point to is that it really is a visionary goal. And it is a staged goal. One is going to reach this goal in a series of steps, and the NIH study group has outlined what those steps should be in their view. I think it makes a great deal of sense. I'm not going to take you through that this afternoon. But a lot of the goal involves trying to understand at a very high level how neural circuits work, how individual circuits that are involved in motor activity, for example, or in decision-making, for example, how these circuits actually work in the brain. These are visionary goals. Second, the timing here is really almost perfect. And the reason for this is that the engineering silo and the neuroscience silo have suddenly realized, starting with that meeting at the Churchley Hall in the United Kingdom, in England, they've realized that there are real opportunities here. The bioengineering, as you'll hear from Shu Chien, nanoengineering, electrical engineering, computer science and engineering, these have all, in the past 10, 15 years, come to a very interesting and very high level of sophistication that can now be extremely useful to neuroscience. On the other side, the neuroscientists have realized, embarrassingly, probably for the first time, that they can really take advantage of these new tools and accomplish things with them that they just dreamed about in the past, didn't have the technology. The objectives have to be focused. You have to spell those out. I've mentioned that already. But finally, I think economics, again, is going to be something that is going to really carry the day here. I think the vision is that there will be, as I'll describe shortly, ways in which this will develop new technologies, and those technologies will have high impact on the country nationally, but we hope very specifically here on San Diego. So let me tell you about Moore's Law for neurons. Now, you're all familiar with Moore's Law. Moore's Law states that the number of transistors on a chip will double every 2 years. And this was a prediction that was made now many years ago, but it's been amazing the way in which Moore's Law has stood up over time. And so what we've plotted here are the numbers of neurons in the brain that can be recorded simultaneously as a function of the calendar. And what you can see is that back in 1960, we could record on this semi-log plot here, we could record a couple of neurons at the same time. And I was a graduate student back then, and we thought that was pretty cool. But you're remembering now that we have 100 billion neurons in the brain, so it's kind of small potatoes when you think about it. And then over time here, we've seen that the numbers of neurons that can be recorded simultaneously has increased, and this is good. But notice how good it is. Not very good, right? This is only about 300, maybe 500 in a good day, in a good system. And so when we think about what our goal is, let's just think about recording from 1 million neurons. At this rate, we'd get to 1 million neurons here, 10 to the 6th, at the end of the century. Now, that's really not acceptable. That's just too slow, right? That's just embarrassing. And so the plan is now to speed this up so that in 15 years, we can at least get to 10 to the 6th. Now, some of you are going to be thinking, well, come on, Nick, 10 to the 6th, you're mentioning 10 to the 11th. Yes, true, but if we can record from 1 million neurons that we can select, and we can record from neurons that are in a particular pathway, that are subserving a particular function, a particular behavior, then we think we can really understand how those behaviors are generated. So this is very exciting. Let me, in the last part now of my presentation, talk a little bit about what's happening here in San Diego. And very nicely, we've been able to establish, thanks to many friends, Bob Dynes among them, a Center for Brain Activity Mapping here at UC San Diego. This is something that Pradeep Khosla actually enabled within 6 weeks after the April 2013 meeting in the East Wing of the White House. And my colleague Ralph Greenspan is the director. It's part of the Kaplan Institute for Brain of Mind that I direct. So Ralph and I working hand in glove on this process along with Terry Sejnowski. And we've had a series of objectives that have played out now rather nicely in the year that we've been in business. The first thing that we did was to attack front on the engineering silo and the neurosciences silo and find ways to melt the walls so that these people would begin seeing what the opportunities were to talk with one another. And we did this by getting together at the Qualcomm Institute. It's a wonderful venue, and we had tables and whiteboards and people. We had engineers pitching to neuroscientists, and then we'd have another one with neuroscientists pitching to engineers. And I have to tell you, the engineers had developed these amazing little widgets that could do things that would blow your mind, and they really didn't know how they were going to apply them. They didn't know what to do with them. The neuroscientists, on the other hand, were hungry for technology that would allow them to begin to record and stimulate from large numbers of neurons all at once. So these conversations were very exciting and got people thinking about how can we collaborate? How can we work together? How can I, as a neuroscientist, use your tool as an engineer to achieve some of these goals? This then led to the award of seed money grants here in San Diego. Pradeep Khosla provided $1.5 million to launch this process. These are small grants, really seed money grants, but they provide the opportunity to put an engineer and a neuroscientist in the same room, lock the door, wait for the white smoke to come out, and see what can happen. And we've funded now about 24 of these innovative seed money grants, and I'll just illustrate for you some of the topics of these proposals. So you get a feeling for what's being funded, what these guys think that they can do. So here's a fully encapsulated wireless, modular, and conformable neural recording system that should be able to record and wirelessly, while the animal behaves, moves about, beam the input and multi-channel input back to a computer. Here's another kind of completely different technology involving nanoshells. These are tiny little micron, submicron sized plastic biodegradable shells that contain enzymes that report on neurotransmitter distribution in the brain. We've already talked about synapses and how neurotransmitters are key to the function of synapses. You need to know what neurotransmitters are where, when they're being released, what their distribution is. So this is a very exciting project. Some of you will remember, now this was a long time ago, this certainly dates me, the movie, The Fantastic Voyage. Anybody remember that? Anybody? Yeah, good, okay, right. So this was a wonderful fantasy at the time involving human beings that were shrunk down to submicron size, got into a little submarine that could then travel through capillaries throughout the entire body and be able to report on the status, healthy, unhealthy, of different organ systems. Well, it turns out now that technology can probably arrive. It's possible now to make very small robots that can travel through blood vessels, that can be recording the activity, the chemical, the electrical activity in the brain, and also can be recording activity in the liver and in the pancreas and in the lungs and in other tissues. So this is going to be an amazing technology when it comes to reality. Another kind of approach has been to have these micro pillar arrays of very fine needles that can be pushed into the brain without damaging the nerve cells in the brain to be able to record from very large numbers of cells in 3 dimensions. And finally, optical methods are being developed that will allow through focus capture of the activity of neurons in the intact and functioning brain. So these are just a few of the kinds of seed money grants that we're funding with the Center for Brain Activity Mapping. And then finally, we've convened scientific meetings to actually bring together people from across the country to rapidly disseminate progress. The first of these was again held just this month here in San Diego, June 4th. This was a symposium on sensor technologies for the nervous system. What are the technologies currently available out there that people are developing, that people are using to record the activity from as many neurons as possible in the brain? Speakers including people from MIT, from Harvard, from the Scripps Research Institute here, Sanford Burnham, from Santa Barbara, from Chapel Hill, across the country here, coming to San Diego. Very exciting symposium, lots of interest and lots of further collaboration set up as a result of this process. So this is locally in San Diego. Some of you will have followed perhaps the passage of the California state budget, thanks to Ralph Greenspan's efforts, and again with great political input both from Larry Smarr, from Bob Dynes, and also from Dick Atkinson. This really was very well positioned, and we saw the creation of Senate Bill 836, Brain Research, a cow brain program. So this is not the federal brain initiative, this is the California initiative, Cow Brain, state of California. And at the time that I prepared these slides, all I could tell you was that the budget of $2 million for fiscal 2014 was going to be voted by the legislature on June 15th. That had happened. But very nicely, just last Friday here in San Diego, Jerry Brown, our governor, signed into law this very nice $2 million for CalBrain. So this is now a little out of date. It's not on the governor's desk. It's beyond the governor's pen. And this is going to be spent now to jumpstart a program that will be across the state of California. This is not all going to be based in San Diego, but we believe that this would be the initial funding, just like the initial funding for the BRAIN initiative, that will then lead on next year, if, as Bob pointed out, the economy in the state of California continues as strong as it is, to allow us to achieve even greater support from the state. And here's just some language to indicate what was going on in the Senate bill. The BRAIN initiative has the potential to be a major driver of new industries, jobs in biotechnology, artificial intelligence, information technologies, as well as a catalyst for major breakthroughs in brain-related diseases, injuries, and illnesses of the kinds that Larry Goldstein told us about. So what's the plan for San Diego? We think, and I hope I tried to give you some flesh on the bones here, we think that innovative neurotechnology will be developed here in San Diego. Yeah, it'll be developed elsewhere, but we think we're way ahead when we talk to our colleagues around the country with what we're getting to happen here. We think that as a result of that, companies will be founded. That was the pattern for the Human Genome Project, a number of companies founded here in the San Diego region. We think that will happen for neurotechnologies as people develop these amazing tools. They want to manufacture these tools. They want to disseminate the sale of these tools to neuroscientists and engineers around the world. We think that will happen here in San Diego. One thing that's for sure to happen is that we will be training students. This is part of the educational mission that Bob Dynes talked about. Students that are cross-trained in engineering and in neuroscience, so they can bridge this, what used to be a divide, and now work seamlessly across these different fields. And so finally, there will be jobs here in this sector in San Diego. So we're very hopeful here for a very bright future for this process. I want to close then with just a quick shot. Some of you will know this view. This is the view of the shore, just a couple hundred yards to the west of UC San Diego. Wonderful spot, but I want to highlight here in my acknowledgments to Ralph Greenspan, Terry Sojnowski. I've mentioned their names a lot. Also, Chancellor Pradeep Khosla, been a real engine here for us, driving this process, as well as UC President Emeritus Bob Dynes and UC President Emeritus Dick Atkinson. Thanks very much.
I think we have time for one question for the doctor. Anybody?
Yes, the question is, what's the relationship of the BRAIN Initiative and CalBrain and Center for Brain Activity Mapping to the European BRAIN Project, which is a very big project, very well funded. This is a wonderful project, very complementary to what's going on here in the United States. If I can characterize it briefly, the European BRAIN Project is involved in taking existing data, both functional and structural, anatomical, about the brain, and existing computational algorithms, and trying to reconstruct how the brain works, how a small piece of the brain would work. We think this is a very important goal. What we're doing here is trying to acquire additional data with these new tools and develop new computer algorithms to go beyond that. We will build, in part, on some of the things that the European BRAIN Project have already developed. Great question. Yeah. Good.
Welcome back, everyone. I hope you enjoyed some of the fabulous San Diego weather there. I can appreciate it, especially being from Chicago this past winter. We're moving on now to emphasize the interdisciplinary nature of research in neuroscience and the other subjects that have been mentioned today. And near and dear to my heart, Dr. Shu Chien, who is celebrating his 16th anniversary here at UCSD, is an engineer and has focused research on bioengineering. Dr. Shu Chien received an award from President Obama for the research, and focuses research on the physics and the interactions and pressures and the biological response in cells. And I'm very happy, and I'm sure you will all be happy to hear about the important research being done by Dr. Chien's lab.
Thank you very much, Steve. Good afternoon. It's been a wonderful meeting so far. I've heard the marvelous talks by Bob Dynes, Larry Goldstein, and Nick Spitzer, bringing out the importance of interdisciplinary research. I think Nick said several times you want to break down the silos between engineering and medicine. That sort of really set it up for my talk. And Larry Smarr, who's going to finish the talk session today, he's been doing that really very actively, including on himself, personalized medicine with engineering. So this is really a wonderful time for us to get together to talk about how to how to catch the next big wave. So I'm in the departments of bioengineering and medicine, and also the Institute of Engineering Medicine at UCSD. What I'm going to talk about is not what I have done in pressure flow effects on endothelial cells, but rather to talk about the wonderful work my colleagues have done in the Institute of Engineering Medicine and others who can illustrate how engineering can be applied to medicine to improve healthcare that would benefit people. Where the big wave was made at the turn of the century when the human genome was sequenced, and that opened up a lot of new vista for us to follow. And with that, we have so much data, and it's accumulating more and more all the time, and we need to be able to integrate this kind of information. And integration here, we need the integration of biology, medicine, and engineering. Of course, other sciences, chemistry, math, and other sciences, and to integrate across different levels in the biological hierarchy, from genes to cells to tissues, organs, to the whole body, and even the society. And then we need to integrate education, research, and also the technology and translation. So we have to work with industry, and we have to work with our clinical colleagues for the translation of our research results to benefit people. Talk about the biological hierarchy. As I mentioned, we can go from genes to molecules to cells, tissues, organs, and systems. And even the systems and the genes are related because genes can be studied by using system biology, and what happens in the system can be studied by manipulating the genetic makeup. And I'll start by talking about the nanotechnology, which Nick already referred to several times in relation to neuroscience. And here I'd like to talk about the work that has been done by my colleague in UCSD. Is there another pointer somewhere? Should be right on the podium. Right on the podium? Oh, here it is. Thank you. In delivering nanoparticles with drug payloads, the one thing we have to deal with is that these are usually foreign to the body. They are recognized as being foreign, and they cannot do what we would like them to do. So what Larry Smarr has done is to use the native cell, the red blood cells. You and I have a lot of them floating around, and what he did was to lyse them to take out the content and use only the membrane, which then has various kinds of proteins and lipids in there, but without the interior in which you can package the nanoparticles. In this way, you can deliver the nanoparticles to various tissues and cells and do their job. It's a camouflaged nanoparticles so that the body will not reject that. You can deliver therapeutic agents, deliver imaging agents, And also you can use it to absorb toxins by using the surface of the membrane, because the rest of the membrane can do that. So this is a very innovative approach that make us possible to use the nanoparticles much more effectively without many of the side problems. Besides the red cell membrane, he has also used the cancer cell membrane. Here's a cancer cell which has been lysed, so you have only the membrane without the interior, without the nucleus with the genes and all the proteins in the cytoplasm, but only the membrane. In the membrane, we can package the nanoparticles in there as the core, and this is the cancer cell-coated nanoparticles, CCMPs, and these can be then used to deliver antigen to antigen-presenting cells to work on immunological approaches, and also it can then use these to link with the cancer surface molecules. We call it homotypic, that is, same molecules will recognize each other, so we can deliver the payload of treatment particles to the cancer cells by conjugating them with the same surface molecules, so they can get specifically to the tumor cells. One of the important things for treatment is not to get it to the regions that are not desired. We want to get them to the region we want, that is the cancer in this case. So this will really allow us to do very specific presentation of the therapeutic agents. This is the work done by another colleague, Ada Amatari, and what she has done is also on nanoparticles, using these hydrated polymer nanoparticles, in which we have confined water encapsulated payload within this polymer nanoparticles. And then you can irradiate the nanoparticle with infrared radiation, which will have heat transfer to the polymer matrix, and this photothermal heating will make the polymer become rubbery and release the water. So water becomes free, and then the payload can be delivered. So that this is a target delivery. You can shine the infrared at desired location, so you can deliver the agent where you want it. So these two are, now this method is not only good for nanoparticles for cancer treatment, but it can be used for a variety of other kinds of applications by using photoactivation control, the chemistry in time and the space. I'd like to move up to the tissue level. Tissue engineering is an important topic. We deal with cell interaction with the matrix on which they sit, and interaction between cells and cells. And Larry has given an outstanding talk on stem cells. I'll just show one slide. Of course, you know that stem cells, pluripotent stem cells, can be differentiated into a variety of cells, such as blood vessel, heart, liver, and pancreas. And it's well known that the differentiation and the proliferation of the cells depend on its microenvironment. That is, the physical-chemical properties of the environment. There is a lot of study on the chemical stimuli, such as growth factors and so forth, on the signaling pathway to cause the stem cells to either differentiate or proliferate. And it's also known that extracellular matrix, the proteins here, such as collagen, fibronectin, etc., they interact with the cell to modulate their fate, as well as the cell-cell interaction. What has been shown in recent years is that not only the chemical factors, but also physical factors, such as mechanical stimuli, such as pressure and flow, can also modulate the behavior of the stem cells. And furthermore, the matrix rigidity on which they sit has a very important influence on what the stem cells do. So I'll just show a couple of slides on these two factors, the rigidity effect and the extracellular protein effect. The rigidity effect was discovered by Adam Angler in our department when he was at UPenn as a graduate student. His thesis work showed that if you modify the rigidity of the environment of the stem cell, you can get different kinds of differentiation. And for example, use a very soft matrix, then the cells that has multiple potentials will go into the soft cells, which is the neuron. And you can see this is the markers for the neurons, you can see they show up, but not the muscle or the bone. On the other hand, if you use intermediate matrix, then you see the muscle differentiation, not the nerve nor the bone. Now, if you use a very hard environment, that is, you can see it's more than tenfold over this soft one in rigidity. Now you get bone differentiation rather than the muscle or the neurons. So the cells can sense their surrounding. What the rigidity is, it will become similar to that surrounding. So there is a continuous interaction between the cell and its microenvironment. The cell can be modified by its environment. In order to understand what kind of matrix proteins can modulate the fate of the stem cell, we have a microarray system. You can see there are many, many of these spots on which we can spot different kinds of matrix proteins. And then we put the stem cells on there to see which matrix protein and their combinations are most beneficial for a particular kind of differentiation that we want, or proliferation. Then we can decide with this relatively high throughput technology what is the optimum environment for the stem cell, either for differentiation or proliferation. This is a higher magnification, and then we get this kind of there's a spotting device, make these kind of array. Then we can look at the array and their stand, so you can get the output from which you can determine the optimum ECM composition, not just one protein at a time, but various combinatorial ways. What are the 2 or 3 combinations that are best for the differentiation? So this will help us to make a large quantity of stem cells and also to direct their differentiation into a particular lineage. This kind of approach we have adopted to use it to determine the anti-cancer drug, because as you know, cancers don't always respond to one kind of drug, but very often they respond more effectively to combinatorial drugs. And in order to study that, it takes a lot of iterations, a lot of experiments, but using this kind of approach, you can spot different kinds of drugs in different combinations. You can put a patient's cancer cells on there to see which one surrounding which kind of drug is most effective for this kind of cell. Now let's move up to the function level, at the organ level, and this is the slide from Todd Coleman in our bioengineering department, and this is the epidermal electronics. He is able to use a very thin piece of material put on the skin. As you can see, it's very thin and very flexible. You squeeze with the fingers, you can see it will deform, but without changing its structural features. You can still function. It's ultra thin, ultra light, ultra low modulus, very soft, and this kind of the electronics are robust to mechanical deformations. So you can use this to study various kinds of signals from the organ or tissue that we're looking at. And in there are sensors for electrical stimuli, for temperature, for mechanical strain, and for all kinds of sensing, and also has the LED system in there, wireless communication, and light sensors, so that it's all embedded in this thin piece of, small piece of sensor, and you can put it on the abdomen of the pregnant mother and record ECG, electrocardiogram from that. Instead of like in the old days, you have to strap up the mother and with all kinds of restraining devices. Now just one piece of this kind of tape-like thing would do it all. And the same is true for the infants. Instead of having the infant strapped up like this, now you just have this little piece here, and you can record all the things you want, non-invasively and very effectively, and this is very, very useful. Now, Nick Spitzer talked a lot about the brain, and I'd just like to show this one case that we can use engineering approach to sort out the brain waves, the electroencephalograph. It originates from different parts of the brain, and they're adjacent to each other, so their waves interfere with each other. So when you record here, it is affected by activity here and there, and there is a way using engineering technique, mathematical modeling, to sort out the signal in the face of many noises. So here is an example of the so-called cocktail party problem using this blind source separation to sort out the noises from the signal. So people are there maybe talking and having various kinds of noises like music of various kinds, and this person is talking, it cannot be heard. But if you sort out these, then you can hear what he's saying. Then people appreciate it and will applaud. So this is a sort of example showing how the brain waves can be sorted out by using this kind of computational approach, so you can get what you want, taking away the noises. And this is so you can get separation of the brain from the non-brain signals, so that this kind of electroencephalograph can be analyzed with more meanings. Now we move up to the systems level. The system biology is a very rapidly developing field in recent years. There's a slide from my colleague Bernard Paulson showing how the system biology brings together biological sciences and system sciences. So from cells to genes, and here your system approach from modeling to mathematical solution. So you bring the two together to have a genome scale interaction network. So you understand how the genes behave in the presence of other genes and other signals. Now, as I mentioned, the turn of the century, we sequenced the human genome, but in 2007, Jim Watson decided to sequence his own genome, and that paid about $2 million. And you saw Craig Venter's picture earlier, and Craig did it himself also, but those are very expensive endeavors, and Science magazine every year selects a discovery of the year or molecule of the year. Then in 2007, the selection was human genetic variation. In other words, instead of having a group, you look at individuals. These genomic variations can make a difference in disease susceptibility responses to drugs. So how we are susceptible to diabetes or hypertension or certain kind of cancer could be obtained. The information could be obtained from the genomic sequence, and also our responsiveness to drugs, whether we're hypersensitive, having side effects, or not sensitive, or just perfectly sensitive, can be determined this way. So it's very important. So everybody wants to know not just the human genome, but my genome. And the target at that time was to have a $1,000 human genome determination of the individuals from the $2 million that you saw. And in 2001, actually, it was very expensive, and 2008 it drops down. In 2013, last year, to 6,000. But Illumina has really led the way in doing this, and they said now it's already below 1,000. It's reached the target, so that we can all get our human genome sequenced if we're willing to pay that, or maybe one day the insurance will do that. But there are also other kind of questions like if you know all of these, is it good or bad? Just like sickle cell disease, when it was first decided to do the gene sequencing, gene detection of sickle cell disease, there was a lot of argument whether you should do that or not. Should the man and woman with sickle cell trait, should they marry or not, all of these effects. If we know too much about ourselves, there are other consequences, so it's not just medicine. There is humanities and social science considerations as well. Now, the genes are very complex. This slide taken from Trey Ideker shows the gene circuitry. The genes are connecting with each other almost like a modern art. And not only the genes, the DNA sequence, the ACTG sequence, but also their epigenetic modifications outside the sequence. They can be modified either by changing the histone around which the DNA wrap. If you change the histone, then this will change the gene expression. And the other is the DNA methylation. If you methylate the DNA at certain spots, you can change the gene expression. And finally, the microRNA, which was which became popular in the last decade, it is another kind of epigenetic effect. So besides the genes themselves, the sequence themselves, the epigenetic factors need to be considered. So we really need a system biology approach to bring all of these together in a meaningful way. So the data is going to be huge, and I'm sure Larry will be able to talk to this much more effectively and knowledgeably than I do, but I'd just like to bring up the big data is a big deal. It's really very, very important because we're getting data left and right, so we need to be able to structure them such as for molecular profiling, phenotyping, for imaging, and so like imaging here, the sequencing here. We need to be able to handle the data using the big data approach. And this is not only important for us in healthcare to capture, curate, store, search, share, transfer, analyze, and visualize data, but this is also can be applied to business, to legal actions, to traffic analysis, and determination of research quality, finally diagnosis, treatment, and prevention of disease. So this big data is really very, very huge in its implications. Now, I remember days I thought terabytes were huge. I mean, the supercomputer center handles that. Our lab has nothing close to that, but that's small now. In 2012, 2.5 exabytes data were generated every day. Exabyte is here. So if it's every day, then a year is already got to the zettabyte, and that's 2012. Today is 2014. I'm sure it's already, Larry will know, whether it's yottabyte. Then what's the next name? I don't even know, but maybe it's invented already. If not, it has to be. But anyhow, we need to deal with this big data, and then need engineering and medicine to work together. Now I'd like to show some kind of integrative results. This is to apply engineering modeling to cardiovascular surgery. This is a vascular anomaly, and we can do the model simulation to see how the flow will work. And here's the X-ray showing the actual operating room picture. Then we can do simulation and even trials of operation without the real operation. We can do the simulation and see what's the best way, what the flow will be like. So this will be good. And surgery is going into robotic surgery, non-invasive, minimally invasive surgery, going through the natural orifices, so we minimize the operation wounds. And there are other ways of using engineering approaches to improve the accuracy of operation, to remove like a tumor as much as we can without damaging the nerves. In this picture, this is the work done by Roger Chan's group, and together the surgeons in UCSD, they can put in dyes to stain the tumors and then stain the nerve, because it's very important not to injure the nerve. Like a prostate cancer operation, if you damage the nerve, you have all kinds of problems with urination and sexual action and so forth. So it's very important to preserve the nerve. So the way to do it is to be able to stain both of them. And Roger developed this kind of stain to stain the tumor with this ACPP and pseudo-colored green, even though the fluorescence was not not green originally, but he's like a magician. He can make it green, and then he can make the nerve also target stained and make it aqua. So you have the nerve stained and the tumor stained. You can switch back and forth to look at the two, so you can remove as much tumor as you can without touching the nerve. I mean, that achieves the two major purposes at once. Now, engineering has helped us in many ways. Here again, we go to a neuro-related problem, and this is the work done at Medtronic. Paul Citron is our adjunct professor, and this is a slide showing how electrical stimulation can be used to treat something like the Parkinson's disease. Here is a patient, and this has sound. They're talking about Swedish, so whether you can hear it or not doesn't matter, but it does have the screen, on the screen, the English subtitle. But so that, oh, sorry, I should have clicked on here. So this is a pianist, very famous. Can you hear it? It's not too loud, but it's okay. So you just look at the title, subtitle. So he was shaking like this, but then they had an implanted electrode in his brain. Oh, now we hear the sound, yeah. I think it's actually Danish. I think some of you may understand that. So when you stimulate him, he can pour coffee, no shaking. So if you put in the right place, it's not easy to put in the right place. So in December that year, he showed in front of the audience and played his piano. He was not able to do it without this. You see, the patients were, the audience was really moved. You see, that's really what science is all about. You can really do so much for the human being, for our health and happiness. And finally, I'd just like to mention that information technology, wireless health is playing a very important part in medicine, health care, and that's, I'm sure, Larry is going to talk about, so I'll just show this slide and without saying anything more, but this is very important. So to summarize, the frontier areas that I mentioned so far include nanoscience and nanotechnology, imaging, genomics, regenerative medicine, system biology, novel surgery and devices, wireless health, and personalized medicine. And overriding principles that guide all of these that we must not forget is that we need to be integrative and interdisciplinary, and we go across multiple scales. We must translate our innovative research to clinical application to benefit a patient. And we have to have low cost effectiveness. Otherwise, everything just gets more and more expensive. We cannot afford that. So the engineers have a responsibility to develop new ways to better diagnose, treatment, and prevention, but with less cost. And equitable distribution of health care to regions that are not as affluent as where we are, and to be able to get this kind of benefit. And finally, we must have integrity and ethics. That goes beyond everything. It must be there. So the healthcare is undergoing tremendous transformation. It's instead of just being in the hospitals, it can be in multiple locations because the wireless approach, you can go to many places, and instead of having fixed appointments, you can do it various times, and instead of treating everybody as part of a population, you can also begin to look at individuals with these personalized approaches. The healthcare professionals, instead of reacting to disease, they will focus on prevention. And the surgeries, instead of being invasive, will become non-invasive. The cost, we like to see, instead of being expensive, but cost-effective. So in summary, it's individualized preventative medicine that is less infrastructure dependent and more patient-oriented and cost-effective. So I'd just like to show this one slide about the Institute of Engineering and Medicine, which I mentioned at the beginning. The IEM, our discoveries are translated to the clinical side through the collaboration of the Altman Clinical Translational Research Center, as well as the various hospitals, and to industry through the collaboration with Biocom Connect and many other organizations here. We're very fortunate in San Diego that we have these organizations that will foster the translation. And then at UCSD, we have the Von Liebig Center for Entrepreneurism, and of course the Tech Transfer Office, the TTO, which is organizing this symposium. I'd like to thank all of them. And so in summary, not only academia has to work hard on these problems, we need to work with medicine and work with industry, and we need to have the society support, the government, and even international. So with all of these, then we can improve people's health and combat disease. And with all of us working together, we can really catch the big wave. Actually, my thinking is not just to catch the big wave, that's passive. We will make the big wave. That's what we do. We'll make, thank you.
Our next and final speaker is Dr. Larry Smarr. He has come here to San Diego from Illinois, and big data was mentioned. He's certainly familiar with big data. At the University of Illinois, he was the founding director of the National Center for Superconducting Applications. Here at San Diego, he is exploring the individual effects, and so much so in monitoring his own conditions and exploring how all this information can be used with bioinformatics and with computing power and Moore's law to increase knowledge, to personalize your own health status, and to perhaps challenge the current model for the healthcare system. Please welcome Dr. Smarr to the stage.
Well, thanks very much. We have an institute, one of the Gray Davis, Governor Gray Davis Institutes for Science and Innovation. In fact, Bob Dynes was, along with Ralph Cicerone, the then Chancellor of Irvine, the co-PIs on this back in 2000. And the UCSD, UC Irvine partnership each have a building. The one here, Atkinson Hall, houses the Qualcomm Institute, which you've heard mentioned. And so what we try to do is live in the future and report back. Some 5 to 10 years, basically. In other words, take the trends that are currently clearly happening, but then go up the exponentials a bit to see what things would be like in a few years, and then figure out how that's going to radically change things. Now, I'm going to talk about one aspect of the things we've studied at Cal IT2, in particular, since the beginning. You remember this $4 billion human genome? Well, at that time, we said where we were going to be now is in digitally enabled genomic medicine. The genome would have gone from $4 billion to $1,000, which it has. The cellular internet would have been built out. It was 3 years later in the future that the first cellular internet was introduced in the United States, and starting in San Diego and Washington, D.C., and that would enable to read out the state of your body. Now, I'm a physicist, computer scientist, and when I look at a complex dynamic system, I used to do, black hole accretion, colliding neutron stars, things like that. You have these complex systems. You look for what the base variables are and then see how they develop in time. That's the only real way you could explain things. So when I got into this idea of quantifying myself, I assumed that's what you do. It turns out that's not really what medicine does, but it's where we're going, because we will now be able to do this, and we never were able to before. This idea of quantifying yourself, not just the way I'm doing it, but lots of ways, is explosively grown. The first meeting of Quantified-Self.com in 2008 has grown now to 116 cities and 37 countries just 4 years later. There's an explosion of widgets that you've seen. I was one of the early users of body media back 10 years ago. I did 700 nights of using the Zeo on my forehead to measure every 30 seconds the state of my sleep. I've done months of doing every gram that goes in my body with these things. These are all things I've used. But we're past that point now. Really, we're in the merger and acquisition and bankruptcy phase of a startup. This is not just something you can ignore. I mean, when Kleiner Perkins leads a Series A, it's time to pay attention. This is taking off. Fitbit, which Bob Dynes and I are among, say, a dozen people who each week report the number of steps we've done. As a youngster compared to Bob, I am humiliated by Bob every week because although I can get like 8,000 steps, 4 miles a day on average, Bob does twice that. So I've got to pick up my game. So the point is that this is becoming a real social phenomenon, so much so that the Robert Wood Johnson Foundation funded us first to do a survey nationally of this field, and now has funded us to actually build a network of innovators across the nation in personal health data. Now, Lee Hood invented that gene sequencer we heard about a long time before the human genome was actually done with it. And he is the head founder and the director of the Institute for Systems Biology in Seattle. And he has been saying for 10 years that what we're seeing is this convergence of this systems biology you heard from Shu about to systems medicine, the consumer-driven social networks. We talked 10 years ago, and you say, yeah, well, how different is the world 10 years ago? Well, 10 years ago, Zuckerberg was an unhappy male in a dorm room at Harvard whose girlfriend had just done something bad to him, he thought, and so he hacked into all the other dorms and got the girls' faces and created the first Facebook. There are now 1.3 billion people in the world, and Zuck, his net worth went up by $2.5 billion in one day a few months ago with just one move of Facebook stock. This is how rapidly the scale is changing. And when you intersect that with systems biology and the big data that Shu talked about, you get something that's very different. Now, I, for years, because of my quantification I'll talk about, Lee Hood has referred to me as his human lab rat to sort of see the future of this. But the question really is, as I become more common, how does the qualified consumer integrate into the healthcare system? Because all this data is outside of the healthcare system. It's not in your medical record. It's not something your doctor knows. And it's exponentially increasing, whereas what's in your medical record isn't. So Lee has started up what he calls the Healthy 100 Pioneer Program. I'm one of the first people in it. And this is not just doing the self-tracking devices and medical histories and so forth, but taking and preserving blood, urine, and saliva, looking at not just your human genome, but your gut microbiome, which I'll talk more about. And we're starting with 100 people, and then we're looking at them in time over time. It's like a Framingham study. So the idea is to take people who are healthy, but a certain fraction are going to get unhealthy. Well, Lee believes you'll see that early if you're tracking these biomarkers. Now, I like to say, people say, well, that's what you have a doctor for. Really? One of the things about being an astrophysicist is how many hours in the year there are. And that's how many. And so out of 8,760, it's actually 36. But anyway, one of these, maybe on a good year, you have with your doctor. All the other 8,059, you're alone, on your own, in charge of your body. Every bite, every sleep or not. And so that's why you've got to have tools. If you're going to understand and become a real partner with your doctor, you've got to know who you are. And so here's an example over the last 10 years of just me. I started when I got here, I was overweight and needed to get exercising and so forth, so I started stepping on a scale. So I had one number every day that had a time series of that one number that defined me. But then I realized I could start taking blood variables. I'll spare you the story why, but I started taking 100 or 150 things in my blood and then later in my stool. I was an early adopter of 23andMe, which measures 1 million of the points along your 6 billion diploid human genome. That is where 90% of all human variation occurs. So now I got 1 million numbers defining me. And now that I'm working with Craig Benner's Institute on sequencing my gut microbiome, I'm up in the tens of billions. And so this is what you're going to have for every individual. Now, how do you go through this big data? Well, one of the things that we've worked at the Qualcomm Institute for many years on is scalable visualization. So you have visual data analysis. This is our 64 million pixel wall. Each one of these screens here is an HD screen, like you'd love to have at home in your home theater. And those graphs are my probably 60 blood tests and 50 stool tests in the last 5 years that are showing you the oscillation of everything like your cholesterol and your liver and your kidneys and your white blood cells and red blood cells and all the rest of it. Well, when I looked at it, I found there was one variable that was way out of whack that was my overall inflammation, my CRP, complex reactive protein. I couldn't figure out why that was and traced it down to just 6 inches of my sigmoid colon, got an MRI, working with my doctor, and then as soon as I got out of the tube, I said, give me my data. That's something you learn to say. Give me my data. It's your data. If your body generates the data, it's your data, and you should own it. And so I took it back because we have, of course, virtual reality experts at the Qualcomm Institute. And so they created this sort of fly-through video game of my body, which I've given many, many tours of now. But you also hear about, you know, these 3D printers. Well, we've been doing that for 5, 6 years. And so I said, well, just send my disease part of my colon to the 3D printer. I said, really? I said, yeah, I think it'll work. And sure enough, there it is. So I can actually go in and look at it and understand it a lot better, and I use it actually with the doctor. But once I understood it was my large intestine that was the source of this inflammation, which was a surprise to me, I didn't expect that to happen, I tried to understand what was it about the large intestine that was the problem. Well, over the last 5, 6 years, we've understood that in your large intestine are 10 times as many cells as in your entire human body. So 90% of the cells in your body are not human. They're microbial. But more importantly, if you talk about the genes, each gene makes a protein on the DNA. And if you say, well, let's think about the DNA of my body, 99% of those genes are not in the human DNA. They're in the microbe DNA. Now, the problem is that none of this microbial information is in today's medicine. It's not in why we have health, we have disease, there's no treatments, anything else, because until we had 100,000 to 1 million-fold decrease in the cost of genome sequencing, you couldn't tell who you were. And this microbe population changes every time you eat something. So the inclusion over the next 5 to 10 years of this vast new trove of information about each of us is going to radically change medicine. So to understand how it might be that that would change medicine, I decided to turn my body into a genetic observatory, by the way, publicly sharing the data, which is a sort of a high-risk behavior when the regulations, as we heard earlier, aren't in place to protect the individuals, except for the human genome information, and even then, only for health insurance, not life insurance, catastrophic care insurance, so forth. A lot of work to be done in the legal and regulatory framework. But nonetheless, we got to move on to figure out what that world's going to look like. So I took, over a year and a half, I took stool samples, sent them off to the Venner Institute in Maryland. They sequenced very deeply using Illumina next-generation sequencers. You come up with basically the unit that comes out of the Illumina is 100 bases, 100 A, T, C's, and G's in a row that you know. Well, it produces 200 million of those. Okay, so you have 200 million jigsaw pieces that you have to put back to figure out the 200 different species, each with their own DNA, that are inside of you. I then was able to go to the National Institutes of Health, which for the last 8 years or so has funded the Human Microbiome Program, and I could download 255 healthy people's microbiome, the data, and also some people that had inflammatory bowel disease, which is what it turned out that I had. And so altogether, we're talking about 2.7 trillion bases of information. Well, to analyze that, fortunately, the San Diego Supercomputer Center here, I was able to get a discretionary grant of 25 CPU years. And so using a very complex set of software that we developed over the last 8 years under the Gordon and Betty Moore Foundation grant to build a global environmental microbial DNA base, we were able to reduce that to well, probably several thousand species, but in you, we look at, say, the top 200. So what you're seeing here is on the left, you're seeing 3 different times, 3 different stool samples for me, and so you're seeing how the microbe ecology evolves with time, and on the other side, you're seeing what a healthy person looks like and what people that have Crohn's or ulcerative colitis look like. And when you put it together, you don't need to know the scientific name, just think blue and red. But on average, people have 2 major phyla, the big divisions of life, that are in balance with each other. And part of the job is to keep suppressing the bad bugs that you all have, but they're maybe hundreds of % or something. But what you can see is that the blue on the healthy gets reduced to a tiny little sliver from, say, 2/3 or 3/4 of your microbe species are in this one phyla to only, say, 1.5%. So it's a drastic change, and that hasn't been really appreciated before, that it's such a big change. Same with ulcerative colitis. You see this explosion of the proteobacteria, which are the E. coli. And where am I? Well, I have features of both. And what this is leading to, I also went through my human genome, and I have certain characteristics that are associated with each disease, ulcerative colitis and Crohn's. So we're going to get a much finer gradation of the types of disease state that you're in. Now you say, well, this is, CPUs and everything else, this is not about consumers, but it is. UBiome, which I serve in the scientific advisory board for, was crowdsourced, funded, and now is sending out thousands of these simple Q-tip studies that you do. You put it in a little vial, put it in US mail. I just did 3 this morning. And this is from July of 2013 until February of this year. The yellow bars are where I did actual samples, and you can see the change over time of your microbial ecology. This is a startup. So the New York Times, when the big results of the Human Microbiome Project came out, said that what we've got to do is get away from this war metaphor that we've always had for medicine, where you're going to kill the microbe. Most all these microbes are incredibly important to keep you healthy. They do all kinds of ecological services for you. So get rid of the war metaphor and talk about gardening. You've got this complex, you are an ecology, just like your garden. And you've got to learn, if you want more of one thing and less of another thing, how do you pull out the crabgrass and make the roses bloom? Well, we have a series of fertilizers, and in this case, I think you're going to see the rise of a tens of billions of dollars a year medical food industry that is tuned to these individual microbes and will bloom one more and get rid of the other, and this is all going to happen in the next 5 years. I think it's just going to be explosive. So everything I've talked about you can do with startups. You can do with things that are under $100 per person, whether it's tracking the blood variables over time, the stool over time, the genetic variations in your human, the human SNPs or your microbiome and its change over time. So then people say, finally, okay, say that I'm an individual, I've got all this data, it's fine for you to be a scientist and you can read hundreds of scientific papers and figure this all out, but what am I going to do? Well, Watson, that IBM developed, remember Jeopardy? It's investing $1 billion in Watson. Watson's read all the medical textbooks. It's read all the scientific articles, and by the way, remembers all of them perfectly. So that is where we are now. But how many of you saw the movie Her? Okay, a lot more of you need to get out. This is an incredibly important movie. It's a good movie, first of all, but it really gives it gives you a human sort of sense of where the artificial intelligence and deep learning based on deep data is going. It's going incredibly fast, and that's why you see companies like Facebook and Google and all the others getting the very best people out of Stanford and Carnegie Mellon and everything else into their companies. Ray Kurzweil, who wrote the Singularity is Near, is now one of the directors of engineering at Google. So this is going to give personal assistants that are always with you, that learn every time you do anything, you eat anything, you move, your heart moves, and it will tune its analysis to you as you give it more and more data, but it also has all of the rest of humanity's data that it's able to compare you with. And so you will not have to worry about having personalized assistance. So in summary, what you've got is, like it or not, there's this explosive growth of data about your body, who you personally are, outside of the medical establishment. You've got the development of what I call these post-Watson personalized coaches. You've got the doctor who now, imagine me going in to see a doctor. It's not like I have a stack of WebMD articles that I printed out, right? This is the new reality that the doctors have to learn to live with. The best doctors you already see are moving rapidly toward becoming a partner with the patient. Coaching has a tremendous experience of cases they've seen before that you will never have as an individual, so it's a tremendous partnership. I think you're going to see a major move toward medical foods from pharmaceuticals, although they'll continue to develop. And with this data, the whole point is to keep you healthy by giving you day-to-day, moment-to-moment feedback so that you make better choices. And the obesity epidemic that's run for 3 or 4 decades in this country will maybe peak and begin to decline. Otherwise, of course, it'll bankrupt the country. Thanks.
Do we have any questions?
Yeah, so the food intake is the most important thing, actually. MyFitnessPal, we just had the CEO, we did a national summit of Quantified-Self at the Qualcomm Institute a few months ago. The CEO of MyFitnessPal was there. 60 million people are currently using it to measure, if you eat processed foods, which I would advise you not to do, you just use the barcode and it uploads all of the data about that particular food. I actually, since I do everything from scratch, organic, locally sourced, wild, etc., I measure every gram into the database. It then breaks it back out into calories, proteins, carbs, fats, salt, starch, fiber, and then you set goals for what you want to modify your body to, and then I did that for 4 months for every day. I've then gone back and did it another month earlier this year. But there's 60 million people using it. You really do build your precious temple one mouthful at a time. So if you don't know what you're putting into your tank, Would you put water into your gas tank because it's over $4 to buy gas? No American would do that. But they throw all this other pseudo food into their body and think there won't be any effect. That's crazy. So we're going to learn better.
I'm just wondering, hearing this is fascinating, thank you. I'm just wondering, as we take this huge amount of data and then we graft it onto into the traditional medical paradigm, we're assuming that a lot of the conclusions of that medical paradigm make sense. So I wanted to raise the issue that's come to me as a practicing oncologist. That, for a while we were treating anemia in cancer, and we came to see it as possibly a good reaction of the body.
The body's fighting, right?
The body fighting. The same might be said of fever, of course, and it might be said of that colony that's causing inflammation within you.
Right. The body makes inflammation because it's trying to do something. I mean, when I measure inflammatory variables like lactoferrin and calprotectin and so forth in my colon, those are antimicrobial inflammatory agents that the body is producing because it detects microbes that it thinks shouldn't be there. The trouble is, when you have such a massive alteration of the expected ecology by the immune system of your microbiome, it goes crazy and starts attacking you. It's collateral damage. And so if we can get the microbiome gardened back, we may be able to turn off some of these inappropriate immune responses.
If I may pursue this, I'm wondering whether that is an adaptive response. In other words, that's part of rebalancing.
Well, it's trying to rebalance, but the trouble is, if you actually look at these curves, what happens is, because it's a nonlinear dynamic system, once you're out of a little local equilibrium homeostasis that you spend most of your life in, and over into one of these other equilibriums, like where I ended up, you know, that's not necessarily a healthy equilibrium, and so the body can try to, you know, use its feed forward and feed backwards, but it's not going to go too healthy. And so there's got to be something that bounces you back over into the healthy equilibrium, and we don't really understand that at all at this point.
Thanks.
Thank you. It was really fascinating. I have a question regarding using the biome information to educate other professionals, such as nutritional experts and registered dietitians, because they're going to have to be educated pretty fast as far as what the public already wants to go into, managing their own health with all these informatics they can get out from the public. So do you have a strategy for reaching out to other professional bodies so that they're moving in sync with you? Because this biome principle is really important for the society to move towards wholeness concept of disease prevention or modification.
So how do we get the word out about the biome? Well, you know, it was the cover of The Economist 2 years ago, the cover of Scientific American, the cover of Science, the cover of Nature, New York Times. I mean, it's getting out there, but the trouble is that the doctors are completely untrained. And they couldn't be trained because we didn't know this information. And this is what happens when you have a scientific revolution of this scale. There's such a lag. I just gave a talk to all the graduating pharmaceutical students on this. You know, I probably was the first lecture they heard. So this is why I don't, it may be that the medical community magically is going to find a way to react quickly enough, but I don't understand what the mechanism will be. This is going to be out in the public far beyond what it is inside the medical community, and that is why I call it disruptive. Now, we see that again and again. That's what disruption's about. So we'll end up with some different involvement, much more paired together, much more wellness focused and so forth. I couldn't predict that at all. I do give a lot of talks like this, but it's such a vast universe out there of the medical field that that's why I think it's going to be a disruption. It's not going to be smooth.
Okay, thank you everyone for coming today, and I believe that you'll see that the wave will continue for quite some time here at UCSD. I will allow Jane to close the ceremonies today before we have some refreshments.
Yes, so just to close the program out, I would like to thank our introductory speaker, Bob Dynes, our moderator, Steve Fallon, and our 4 outstanding speakers this afternoon for an extraordinary discussion of the future of biotechnology. And again, I'd also like to thank our sponsors whose support made this event possible, Greer, Burns & Kane, Perkins Coie, Gravilovich, Dodd & Lindsay, Kilpatrick, Townsend and Stockton, Sugary, Neon, and Sutherland, Aspel and Brannan. I'd like to thank the staff of this beautiful new San Diego Central Library for all their assistance. We're very proud of this building. I hope everyone will have a chance to look around. And last but not least, I'd like to thank all of my colleagues in the Technology Transfer Office and our colleagues in the university's Special Events and Protocol Office for spending so much time and effort to make this symposium a success. Today's presentations have demonstrated why San Diego ranks as one of the nation's leading centers for biotech innovation. And now our closing reception will serve up proof that San Diego is also one of the nation's top regions for craft brewing. So thank you very much for taking part in our Catch the Next Big Wave symposium. And please join us at the reception that features locally brewed beers and appetizers. Thank you very much.